about nucleotide and ribosynthesis. I'm going to just give a quick tour of my board now that it's complete. So we'll start up here and kind of work our way over to the nucleotides so that I can then talk about those. I got the citric acid cycle down here, glycolysis, sugars, and then glyconeogenesis, and a couple more slides over here. I know I'm going quick, but I want to make sure I just cover this stuff to show that it's complete before I start talking about this one in specific. So I'm going to talk about nucleotide synthesis first, and this can happen in vivo or in vitro. So the in vitro is going to be in a laboratory setting, and a purified nucleoside is protected to create a phosphoramidite, which can then be used to obtain analogs not found typically in nature, or if they're synthesized by an oligonucleotide. And in vivo, which is in the body, your liver works to synthesize it, and it also recycles it through salvage pathways. And the components are synthesized and derived from biosynthetic precursors of carbohydrate and amino acid metabolism, and also from ammonia and carbon dioxide. So the de novo synthesis of pyrimidines and purines follows two separate pathways. Pyrimidines are synthesized from aspartate and carbamoyl phosphate. And here's just my little aspartate guy. There he is there. And you can also see here, all the enzymes present: ribonucleotide reductase, CTP synthase glutamine, dihydrofolate reductase, and you can see ATP is the energy source used there, and ADP comes out while uridylate kinase, and my handwriting, thymidylate synthase are working to produce those precursors. So purines, on the other hand, are synthesized from the sugar template onto which the ring synthesis occurs, and the synthesis of the purine and pyrimidine nucleotides are carried out by several enzymes, kind of like I just mentioned, and it occurs in the cytoplasm, not within a certain organelle, but outside of that. And nucleotides undergo the breakdown of useful parts so that we can reuse and synthesize new and better nucleotides should others be damaged. So I'm going to kind of just skip over here. Not talking about glycolysis exactly, but ribose synthesis. The way it's produced kind of is parallel to glycolysis. And ribose is a sugar made by the body from glucose. And it's really important when it comes to the synthesis of RNA and DNA, and also with ATP. We can see ATP over here. But most of the ATP we have is produced by cellular respiration, and made by oxidative phosphorylation. And the energy is released in the consumption of pyruvate, which I have my pyruvate over here. Sorry, I lost my place. The energy is released by consuming pyruvate and being used to create chemiosmotic potentials and pumping protons all across the membrane. I actually have a little picture here that I'll sneak in here in my video. So you can see kind of the overhaul here, just simplified. And ATP itself, this is kind of just a big generic one, but ATP, where's my ATP slide? ATP is the nucleotide that consists of three main structures. It's going to have a nitrogenous base, adenine, and ribose. And the chain of the phosphate groups bound to the ribose. And the phosphate tail of ATP is the power source for it. And the ATP synthase, here we go, that's where we're going, is the enzyme that creates the energy storage molecule, ATP itself. So it's the energy currency of most cells, and so this is how it's produced here. We've got our subunits, the mechanism itself, and then here is a photo, and you can see, again, ATP is the main energy source for all cellular life. And it is a triphosphate where high energy bond attaches to the third phosphate group, and the bond is really unstable, and when it's hydrolyzed it releases a substantial amount of free energy. And not only does it provide energy, but its other essential roles include having the one of the four nucleotides required for the synthesis of ATP DNA replication, and protein synthesis. It also regulates certain biochemical pathways. In mammals, it releases damaged cells to elicit pain responses, and in photosynthesis, it helps to drive carbon fixation. Because it's unstable and it's used for most processes in the body, each cell in the body must constantly produce more ATP or you'll essentially die. I know muscle function is highly dependent on ATP production, which is why people go into rigor mortis. And in total, the organism's requirement for ATP is significant. The average body generates over 100 kilograms of ATP daily, and ATP synthase is the prime producer of ATP in all cells in the human body, that is, and catalyzing the combination of ADP with inorganic phosphate to produce ATP.